I said before, we had an advisory board that was involved in the project, and we invited some of the members of the advisory board as invited speaker for this conference. And so we asked them to give a lecture that will be, as you see, a little longer presentation than the standard one, because they are experts in their activity. And so we have Professor Francisco Vilaplana, who is an associate professor and head of the division of glycoscience at the KTH Royal Institute of Technology in Stockholm, Sweden. And he leads the research group of biotechnology of carbohydrates from biomass. So very relevant to our project, because you know, chitin and chitosan are polysaccharides. With 10 years of researchers funded by public, industrial, national, and European organizations, the vision of his research is to exploit the rich molecular diversity in terrestrial and marine biomass towards novel functional products for food, biomedical, and structural application. From June 2022, he joined Oatly Food Science part-time as head of plant structure and health, where he will lead the research program linking oat structure and the derived health benefit. He is currently the director of KTH Food, a multidisciplinary research environment for sustainable and healthy food system. He is a chair of the panel Bioresource Technology, Chemical and Environmental Engineering of the Swedish Research Council. He is a honorary associate professor at the Centre for Nutrition and Food Science, University of Queensland in Australia. Then he is president of the Association of Spanish Researchers in Sweden and of the Network Association of Spanish Researchers Abroad. So we welcome Professor Francisco Vilaplana. We really are glad that he accepted our invitation.
So thank you very much, Professor Cinelli, for the very kind invitation to be here today. For me, it's a great pleasure to be in Pisa for today. I'm really happy because not only I am meeting old friends, but also I hope I will meet new ones here. So as Patricia said, I am Francisco Vilaplana. I am head of the Division of Glycoscience. And in our department, we are 30 people. The name says it all. We work with carbohydrates, both from the molecular biology, so how they are synthesized in nature, but also we are looking into the structure in biomass tissues and also developing different processes on how to utilize them in the context, of course, of circular economy and bio-based economy. And our target areas of application are structural materials, food, and biomedical applications. So in my group, we've been working for a long time, mainly with plant-based biomass, but also we have been working in the context of other European projects, for example, with fungal biomass. But what I will talk today about is a very long collaboration project. We have been working with this for actually now 8 years, together with Lantmännen. You can see the logotype here. So Lantmännen is the largest Nordic provider of cereal grains. They are based, of course, in Sweden, in Denmark, in Norway, in Finland. And of course, the main crop in the Nordics is actually wheat. So we've been working a lot with wheat bran. And then, as you can imagine, when we are making, when they are making flour from wheat, there is this layer, the bran, which is the outer layer of the kernel, which is unfortunately not much well utilized. This is kind of very similar approach to the ECOFUNCO in this particular case. And this is because the bran, despite we know that it's really good for our health, but the properties, it's very bitter, it's not soluble, so it's very difficult to incorporate it in functional food products. So what we've been doing is try to valorize this wheat bran and try to get high value added materials, and you will see today how we have applied green chemistry and enzyme technology to be able to specifically valorize that biomass. But first of all, we need to understand how the wheat bran looks like, what molecules do we have in there, because that will actually give us the key on which processes to utilize. And here you can see a picture of how the wheat kernel looks like. This is the endosperm with the starch granules. Here you have the different bran layers, the aileron and the pericarp. And there, as I was mentioning before, we have very good and very functional and nutritional molecules that we want to extract. Mainly, you see them here, arabinoxylan, you will be hearing a lot about it in this presentation, and also beta-glucans. The problem is that in the bran, they are very tightly interconnected with other components, and that makes it very difficult to isolate. So let's take a deeper look on how this bran structure looks like at the molecular level. So here you have a molecule of arabinoxylan. So it consists of a backbone of xylose. Here you can see it in orange, that they are substituted with arabinose in different positions. I won't go in detail about that. But the funny and the very nice thing about this is that they are also actually covalently modified by ferulic acid. Ferulic acid is a polyphenol. It's a phenolic molecule. You can see it here in purple. And funny enough, in the source, they are actually cross-linked to each other by diferulic bridges. That's what actually makes the structure of the bran cross-linked and very recalcitrant to isolation. But that's not only the problem there, because through these diferulic bonds, they are also connected to lignin. They're also connected to structural proteins. We have cellulose microfibrils. We have beta-glucans. We have pectins. So that creates a very complex three-dimensional structure that makes it very difficult to work with. So traditionally, in order to isolate this arabinoxylan, alkaline conditions are used. So these alkaline conditions, what they do is to break up this linkage between the arabinoxylan and the ferulic acid. That works well. We get high yields of arabinoxylan. The problem is that we are losing the ferulic acid. And we want to keep it because it has very nice properties. It's antioxidant, it's radical scavenging, and if we eat it, it actually gives us health benefits. So what we did was to actually try to replace this alkaline treatment using new technologies, using green solvents. In our case, we use subcritical water, which is basically water at high pressure and high temperature that keeps the water liquid. And then we also used enzyme technology in order to isolate these arabinoxylan molecules. So you can see here the process is very straightforward. We start with the bran. We do a pretreatment. It depends on what we want to do. We have tried, for example, destarching. We've realized that's very important to remove the residual starch. You can incorporate a treatment to remove the proteins if we want to isolate them as well. But then you can see here, then we subject the bran to this high temperature, high pressure water treatment. We get polysaccharides and oligosaccharides. We can separate them if needed by membrane filtration. And then, as you will see later, we can also apply enzyme technology to get even more out of the residue. So now I will explain a little bit more out of this process. And why do we want to use enzymes? Patricia actually mentioned it a little bit in her presentation, but we believe that enzymes are the nature's catalysts, and they are very environmentally friendly. They work at mild conditions, normally in aqueous conditions. So we believe that they are very, very nice catalysts to be able to both cleave molecules and linkages and bonds, but also to functionalize our different molecules. And I will show you in this particular presentation these two examples, how we can use enzymes to cleave some bonds in particular places so they can actually help us to release more of the biomass, but also to functionalize different polysaccharides to upgrade the biomass and give new functionalities. So well, this is a cartoon that we actually draw, trying to show how this enzyme platform can help us in different contexts, both to break down but also to functionalize biomass. So just a little bit on this subcritical water process. What we did through different projects is to try the effect of temperature, the effect of time, the effect of pH. But we could see is that time and temperature are very important. And we can see here that this is how we evaluated the composition of our extracts through times. And then you can see here in green how we are enriching arabinoxylans, so xylose and arabinose, when we expose our bran to longer times. So in our bran, first we're extracting the starch, the one that is residual in the biomass. Then we extract the beta-glucans, and in the end we get the arabinoxylans. And we can actually reach high yields and high purities, and we have arabinoxylan with a low substitution, which influences solubility. I will talk about that later. And also another important thing about this subcritical water process is that we keep this ferulic acid covalently bound to the arabinoxylan. And this, as I was mentioning before, confers antioxidant properties to our extracts. And here we just wanted to show you how we compare the scavenging activity. This is ascorbic acid and ferulic acid in free form. In green, they are our extracts, so they have quite potent radical scavenging properties. And here, very small in red, you can see the extras using alkaline. So you can see how we really maintain these antioxidant properties. And we can reach in wheat bran contents of ferulic acid between 4 and 12 milligrams per gram. So just 1%, but it actually gives a lot of antioxidant potency. And we've exploited this process to all sorts of different biomass. We've tried with cereals, we've tried with fungi, we've tried with algae. It really works very nicely. But what is really interesting when we actually compare different cereal sources is that we can really tune the molecular structure of our arabinoxylans that we extract. I'm not going to go in detail through this because it's quite of a complex slide, but in the context of another European project, Barbara, we applied this process to corn fiber. Corn is very interesting for us because you can see here, this is a mess. There's a lot of linkages and different functional groups, but it actually works very well to use subcritical water with corn. And as you will see later, we reach ferulic acid contents of almost 40 milligrams per gram. So 4% is actually phenolic compounds, which, as you will see later, gives us very interesting properties when we're making materials out of them. And we can also control the molecular weight. So we're extracting these arabinoxylans in polymeric form in molecular weights around between 100,000 to 500,000. So it's actually quite large polysaccharides. But we were not happy with that completely because when we did our subcritical water extraction, we normally get yields of 60% of arabinoxylan, but there's still 40% that we cannot get out from the bran. So our idea was, can we use enzymes in order to help us cleave this residual arabinoxylan in the residue? So the idea was, okay, we have our first water extract, we get our residue, we do some enzyme treatment, and then we do another cycle of subcritical water extraction to see if we can increase the yields. So together with Novozymes, which is the largest enzyme provider for enzymes for the bioeconomy, we tried different xylanases. So this is the enzyme that cleaves the backbone of the arabinoxylan, depending on the specificity of the enzymes. And we tried 4 different enzymatic families. I'm not going to go in detail about that, but each family has, let's say, specific features on how they recognize the substrate and where they cleave. And we tried 3 different arabinoxylan substrates with different substitutions. So one with very low arabinose content, one with medium, and one with very high, because we thought that that would affect the process of the enzymes. And indeed, that's what we saw. So each enzyme had a different cleavage pattern. Here you can see the activity measured in reducing sugars for the 3 substrates. And making a long story short, the families 10 and 11, basically they're very active, but they release very small fragments. You can see it here from the molecular weight distribution. So this is the control without the enzyme, and here they are the 2 different products from the enzyme treatment. Then we have this enzyme from the family 8 that is very active in unsubstituted molecules, substrates, so it releases linear oligosaccharides. And then the GH5, another enzyme, which is very active on substituted arabinoxylan, it was very active specifically in this substrate. So now we have a platform of different enzymes that we could use in our process. So we selected 2 extreme cases. We selected the enzyme from the family 10. If you remember, it cleaved very much and released very small fragments. And we selected the GH5, which was very active on substituted substrates, but released longer products. We apply them together with another enzyme, which is called feruloyl esterase, which cleaves specifically the bond between ferulic acid and arabinose. And then the idea was to apply another step of extraction after that. And what we could see is that if you see the process without using enzymes, we reach around 30% of extraction yields. But using the different enzyme technologies, we can add up to 74%. We can reach up to 74% of the arabinoxylan to be extracted. So from 60 to 74, so 15% more, with an industrial context is quite significant. But also what is really interesting from our study is that depending on how we tune our process, we can control on one side the molecular weight of our extract, which is related to the solubility and the material properties, but also the substitution pattern. So you can see that we can get a very nice range of different arabinoxylans that they can find each application. So you can see for material applications you want high molecular weight, but for example, if you want to add them as food ingredients that have maybe good solubility and they can reach the gut, maybe you want lower molecular weight and higher solubility. So this is a nice platform, we believe, for material and nutritional applications. So now we were very happy, we have the process, but now we thought, what can we do more with these substrates? And the idea was to apply different chemo-enzymatic modification processes to try to see if we could use these arabinoxylans with the ferulic acid for example, film applications or hydrogel applications. So this is part of the work that Cecile, a PhD in my group, did for her thesis, where we tried different procedures. We tried esterification to modify the hydroxyl groups. I will talk a bit that later, but also enzyme technology again, in this case, to cross-link our substrate. And we tried two different substrates, the wheat arabinoxylan that I was mentioning before, with high molecular weight, 100,000 Daltons, and a moderate content of ferulic acid, and the corn arabinoxylan, the one I mentioned before that is very complex, which has a little bit smaller molecular weight, so 50,000 Daltons, but it has very high ferulic acid content, almost up to 55% of ferulic acid. So, first of all, the first thing that we tried, let's see, can we make films out of this arabinoxylan? Our rationale was, okay, we have our arabinoxylan, we know it has good oxygen properties, but if we keep this ferulic acid, we might have even better antioxidant properties. And we tried this, we made films, but the problem we had with these films is that in the end, the properties were not that good. It was a bit disappointing, to be honest. It still worked well. We could make films with addition of plasticizers. It had very good oxygen barrier properties, but this acetylation somehow didn't work so well in improving the vapor properties, the vapor stability of our extras. But anyway, this was a nice study. I think a lot of work needs to be done to improve the film properties of the arabinoxylan. But what really worked very well was the hydrogel formation. So using enzymes to cross-link, using this ferulic acid that we keep in our arabinoxylan, using lattice oxidation, we can create radicals that can covalently cross-link this molecule. This is what happens in nature, so that's how the bran is actually synthesized. So we can actually sell it as a biomimetic process because we're just mimicking what nature does. So here we demonstrated biochemically that we are making these covalent bonds. So you can see a reduction in the ferulic acid content, and you can see an increase in these diferyl bridges. And we also monitored it by size exclusion chromatography, where we can see an increase in molecular weight and a reduction of size. So we were very happy. We have demonstrated biochemically that this cross-linking is happening. What was very interesting for us is actually to evaluate the functional properties of these gels. So here you can see the viscosity versus shear rate. So this is the soluble arabinoxylan fraction. So when we cross-link it chemically, enzymatically, we can see an increase in the viscosity, which was expected. You can see here the consistency. But what was fun enough is that when we dry it and we resuspend it again, we really increase the viscosity 2 orders of magnitude. So you can see here that we form a self-standing gel. So we were very interested in understanding what was happening with these gels. So we did some solid-state characterization techniques. We did X-ray XRD, so X-ray diffraction, or wide-angle X-ray scattering. And we can see here for our extract, it's a little bit crystalline, 16%. But when we cross-link it, we double the crystallinity of our substrate. So what we thought is that we are inducing this kind of arrangement of the backbone of our polymeric chains. But it was very interesting that when we did this drying and resuspending, we did not increase the crystallinity so much, and we were a bit confused because I was expecting, when you dry, you remove the water, it would increase even more the interactions, but that didn't happen. So then we went further to try to understand why this Yeah, what was the reason for this? And we went to Lund, to the south of Sweden. They're building a huge synchrotron infrastructure there, and we applied, in this particular case, a small angle X-ray scattering, and then we got the answer to our question. So it is not this fine organization at the, let's say, molecular level what is changed, but we are somehow inducing clustering of the chain. So you can see it as a kind of a nucleation effect. So the chains start to even cluster in larger fragments, and that's why we have this huge increase in rheological properties. So we were very happy about this. Now we understand a little bit better how it works, and we're trying to fine-tune this to even get even a wider range of rheological properties. And the last part I wanted to mention to you is we actually applied this same technology to corn because we were very excited about this high ferulic acid content. And this was really nice because just applying the enzyme, we don't need to do any drying. We really get very nice gel properties. So in this particular case, we tried two different enzymes. We tried lacase, here labeled as an L. And you can see here, this is the evolution of ferulic acid content with time. It takes around between 1 hour to 4 hours to get the cross-linking happening. But there's another enzyme called peroxidase that it happens after 5 minutes. So it's like, you can see it as an instantaneous. You just put the enzyme, immediately cross-links. So that was very interesting for us. Here you can see the rheology properties with time. This is again the lacase. This lag time between 2 to 4 hours when the gel properties start to improve. But for the peroxidase, it just happens instantaneously. We couldn't even measure the cross-linking. It happens, very, very quickly. And very funny as well, that the properties are completely different. So we think that this is because, of course, of the kinetics of the cross-linking. We have also different morphologies of the gels. And then what we wanted to see is that, thinking now of biomedical applications or targeted delivery to the gut. So we know that gut inflammation it's a serious disease, and it's normally caused by radical scavenging moieties that happen in the gut. So we wanted to try in vitro with cells from the gut. So what happens if we stress these cells using radicals, and if we put them in contact with our gels, will this contribute to mitigate these radicals in the process? So we compared that with alginate, which is a traditional substrate that is used in biomedical applications. And to make a long story short, here you can see with alginate, we keep a high value of the radicals when we stress the cells. But with our gels, you can see here the complete reduction of the scavenging. So we are very happy about this because also the cells are very happy. They're thriving in our hydrogels. So we see here that we have a good case of proof of concept where we could go forward and maybe contact biomedical companies or food companies trying to see if we can actually use these gels in vivo studies. So that's the end. So I hope I convey the message that it's very important to look at the biomass from the beginning so we understand how it looks like at the molecular level so we can design our processes. And in this particular case, we believe that these feruloylated arabinoxylans are very fundamental and very nice platform matrices so we can develop functional materials for structural, biomedical, and food applications. And also I've shown you how we can use green solvents, like in this particular case, water and enzymes, to create a platform of different materials that can be used for high-value applications. And I hope that maybe through this presentation, maybe a new collaborations can be made because we can actually envision these materials to be used in coatings, for example, or you can actually think of different type of applications that these materials can be used for. So I just want to thank finally, this is my research group, and I want to thank specifically Rescandi, who did the extraction work, Cecile, who's not in the picture, who did the gelling work, and funding from different institutions from Sweden, but also from the European Union and from the Novo Nordisk Foundation that made this research possible. And thank you again for the invitation for me to be here.
So we really thank Professor Vilaplana for this very interesting presentation. We have a time for some questions. Than curiosity. Andreas Mikalski from Mutamaki.
What about the water stability of these gels? If they have humid conditions. Okay. What about the water resistance of these gels? If they get back into humid conditions, will they dissolve again and be degraded?
So we've tried actually swelling of these gels. So basically when we have the the arabinoxylan without being cross-linked, it's soluble in water, so we lose complete functionality. But the hydrogels, as you can see, we can control how much water we get. We can control the gelling properties. So this of course should be studied. We haven't looked so much into that. We wanted to move towards the proof of concept of bioactivity, but that would be something that could be explored because we can tune the cross-linking. So we believe that will modify the water stability.
Thank you so much for the presentation. I have just one question regarding the subcritical water extraction.
And sorry, when you do a question, before you introduce yourself, so all the
okay.
know who you are.
Okay, I'm Adrian Rich from Celabor.
Yes.
Just my question regarding the pressure that was used for the subcritical water, and we know that the condition to reach this critical state of the water is just 10 to 15 bars, but I have seen that you have used 100 bars. Is there a reason for?
Yeah, I mean, we've been working together with Celabor with this process, so you know about it. So basically, the equipment we have in the lab fixes the pressure, so we cannot modify it. So the lab equipment reaches this very high 100 bars pressure. But when we've done some upscaling trials, both at KTH, we have a small reactor of 2 liters, but also in Celabor, in your reactor, pressure doesn't matter. But what matters is that we keep water in liquid state at that temperature. So that's the critical part. As long as we have the pressure, the minimal pressure to keep the water in that state, it works.
Okay, thanks.
So there are more curiosity or question? Okay.
Good morning. Good morning. Before all, I apologize with us with you for my bad English pronunciation. I am not habituated to speak English in public and to speak English at all. But I am, my name is Marco Sandroni. I am a co-worker of Professor Ginelli in another European project called Dogrimas. And in particular, I worked a lot with Bran. So I was a lot interested in some and also with fibrocellulosic materials. So I was a lot interested in your exposition, in particular about some a pair of questions. The first is regarding the structure of Bran and the process from which you obtained the Bran material, no? In particular, according to the process used, we can obtain very specific fractions of Bran. So my first question is regarding this matter. It's not a very interesting matter for an academic issue, but I think it's very interesting for a processing issue for me to understand this first question.
Yes, I can answer to that directly. So, as I mentioned, we've been working with landmen and for these 8 years, so we get the Bran from them. I know that it's a Bran produced by dry processes, dry milling processes. I can't tell you more than that because I always get the batches from them. What we have noticed is that, of course, depending on the process that they use to mill, we get Brans with different, for example, residual starch content. And for this process is very critical. We want to have the Bran that has lower starch as possible. The grain size, or let's say the particle size of the bran doesn't seem to play such a big role. We've tried that as well. But what we have seen, and I don't know if that is because of the process or because of the seasonal variability of the wheat, every time we get a new batch, I'm scared as hell, because we get different properties, and it's very difficult to control. It's very difficult to predict we're going to get this brand and we're going to get this product. So this is something that, as you say, for upscaling, to make this process industrially scalable, that's something that needs to be targeted. But we have not looked into that. That I think that work belongs to the company, if they want to do it, of course. With our help, of course, but I think it belongs to them.
And the second related question, I think I understand well your answer. Is what is the effect on the mechanical properties on the residual fiber, cellulosic fiber, after your hot water and enzymatic and also enzymatic treatment?
Yes. So, well, I didn't show this because it becomes very biochemical.
There is degradation of mechanical resistance of the fibers, or the loss of the lignitic bonds and the other fine structural bonds has no particular effect on the residual mechanical resistance of the fibers.
So I think you're referring to the residue after we extract, or?
After.
So what I can tell you, we have been looking at a molecular level what happens to the residue, because that tells us a lot about how we can improve the process. I can tell you that, of course, we're enriching in cellulose, we're enriching in lignin, we're enriching in structural proteins. So that still has quite a lot of value. We're just starting a project. It will start in September, actually, where we're actually going to be looking at the residue at a closer look and see what are why is it so recalcitrant? That's the question. The mechanical properties, we have not looked at them so much because we haven't thought about that, to be honest. But I think it could be quite interesting to see, because again, it's rich in cellulose, it's rich in lignin. So, yeah.
So really thanks for this very interesting discussion and additional information. We thanks again Francisco for being here.
Thank you.
It's been very interesting lecture.